So good evening, and thank you very much for the kind invitation to come to Toronto. I'm here only for 20 hours. I flew in this morning and I return to the United States tomorrow. So today what I'd like to do is I'd like to talk to you about this amazing technology called CRISPR, which is going to affect not only this generation, but more profoundly the generations to come. And so we're talking here about designer babies. And so I would like to talk about that. And I'm going to divide this lecture into two parts. The very first part, I'm going to go into the science of CRISPR, just because I'm not sure where all of you are with regards to the biology of CRISPR. And then we're going to spend some time talking about the ethics of CRISPR. And one of the things I'd like to highlight is why we as a society, especially in the West, are struggling to try to come to terms with the ethical boundaries, if there are any, on this particular technology. So we're going to begin with the science. And this is a slide that I use when I teach Introduction to Biology, Bio 103 at Providence College, my favorite course to teach, only because the freshmen don't know anything. And it's just wonderful to build. You know, it's wonderful to build the foundations that will allow them to understand biology critically, as well as to process the enormous amount of information that's out there. So when we talk about the genome, we're talking about all the genetic information of an organism. And this is a diagram of a typical human cell, and there are two parts of the cell. So you have the nucleus. This is like central headquarters of the cell. And that's where you have the chromosomes that a lot of people think about when they think about genes. And you and I have 46, 23 from mom and 23 from dad. And if you take one of these chromosomes and you stretch it out, what you get, so this is one of those chromosomes, is you get the iconic DNA double helix. And so what you're seeing here, you know, people don't realize that each one of these chromosomes is actually an incredibly long piece of spaghetti. And the challenge for modern biology is how you get the genome, which is about 7 feet, I'm sorry, I don't know how many meters that is, but 7 feet long. And you're going to try to organize that and put that into a cell that is as large, and the largest human cell is the egg, and it's the size of the period at a 12 New Times Roman 12 font. So you're going to try to take a genome that's 7 feet, and you're going to put it in there, and if you know what it's like to eat spaghetti, there's a chance that you're going to get everything all messed up. And really the challenge for modern biology is trying to figure out how the cell keeps all of this information organized. Now, the way that we portray DNA today is the following. So this is a strand of DNA. This is actually a gene. This is the gene that my students and I have been studying for the past 8 years. It is a gene that we call BAX inhibitor. And the human equivalent of this gene is upregulated. It is overexpressed, it is hyperactive in different tumors. And so one of the things that we've done over the past 7 years is to try to figure out what this gene does. And this gene actually makes a pH-sensitive calcium leak in the ER. That's what it is. It's very cool. It took 7 years for me to say that sentence. And what we're going to do in the next couple of years, we're beginning a small molecule screen looking for drugs that will actually prevent this gene from working, in the hope that we will be able to contribute to the growing arsenal of chemotherapeutic agents. But what I want you to take from this is that DNA can be portrayed as information. And there are 4 different molecules that make up DNA, and they're depicted by each one of these letters, G, A, T, C. And the order of these letters is actually specified by a code. So if you're a molecular biologist, you will know that a gene begins with ATG, and once you see that ATG, you go, oh, there's the capital letter that begins the sentence. And it ends with a full stop, and there are this one here, TAA, that's actually the end, that's called the stop codon, that specifies the end of a gene. And there are 3 combinations of that, TAA, TGA. So these combinations actually help us to decode the information in DNA. So, and you and I have 3 billion of these times 2, 3.2 billion to be precise. 3.2 billion from mom and 3.2 billion from dad, divided into 46 different chromosomes. So if you look at this information, I want you to imagine like an encyclopedia set, where you've got different volumes. You've got 46 different volumes, and the volume is made up of 6 million of these letters. And what CRISPR allows us to do today is you can now pull out volume 14, turn to page 423, the third column of the left side, the fourth sentence from the top, we can change that letter G into an A. And so we have incredible precision now in a way we didn't have. And so this is CRISPR. This is a schematic of CRISPR. CRISPR is a machine. It's a molecular machine. It's a protein machine. It's made up of two components. You have what is called this guide RNA, and that's like the targeting part of the machine. It tells the machine where to go. Which page of which volume do you want to edit? In order to specify that, you need this gRNA. And then you've got this Cas9 portion, and the Cas9, you can think of it as a pair of molecular scissors. And so what this does is that this machine can target a particular location in your DNA, and it makes a cut. Now, this is actually what it looks like. So we've been able to use X-rays to decipher the structure of this machine. And so you have this here. This blue here is the DNA. The yellow is the machine, the molecular machine. And this green part of the molecule is that guide RNA. And so this is what basically CRISPR is about. So here's your DNA. There's your targeted DNA. And so you can target that machine by specifying the guide RNA that is one of the components of that machine. And what happens is that guide RNA targets the scissors, and it makes a cut. And it makes a double-strand cut. It's called a double-strand break. And when this happens in living cells, normally what the cells will do is that they will attempt to fix that break. And one of the reasons why this is the case is because that kind of break is highly carcinogenic. So that kind of break can trigger cancer. And so your cells have protective mechanisms within them that will allow them to fix that break. And in the process of fixing that break, they destroy the information that is encoded in that location of the DNA. So you can actually destroy the gene by simply making a cut exactly where you want it to be. So that's the destruction part. But what we have been able to do now is at the same time that we put the cut, we provide the cell with a copy of the gene that is slightly altered. It's a repair template, and the cell will use that new information to repair the cut. And in doing so, it will replace the original information with the information that you provided. So you can change that G into a C, or that T into an A. And so you have incredible precision with this particular machine. So to give you a sense of the history of this technology, it was first identified at the end of the 80s because there were a couple of molecular biologists, microbiologists, who noticed that in certain bacteria, you had weird patterns of DNA. I'm just going to stop right there. And a Nobel Prize is expected to be given for CRISPR. Now, the reason why it hasn't been given earlier is because there's a pending lawsuit in the United States between MIT and UC Berkeley. And I can guarantee you the Nobel Prize Committee is not going to get into the middle of that. They're going to wait to see how that lawsuit, that patent lawsuit, is resolved before they decide who's going to get the Nobel. So you have the initial discovery of this machine. This machine appears in bacteria, and it's in bacteria. The bacteria they use this machine to defend themselves against the attack of viruses. And what humans have done is biologists have been able to get this machine, tweak this machine, and use this machine to make it an editing machine. And though that basically happens in 2012, so in 2012 there are two competing groups, which is why you have this lawsuit, about trying to take this bacterial system and to alter the system enough so it can change genes in any cell that you can imagine. You want to change the cells in a koala bear, you can do that. You want to change the genes In a kangaroo, you can do that. So we can do all sorts of things now. In fact, one of the really interesting things that people are trying to do is they're trying to change genes in ants. Because what they want to do is they want to figure out how different ants know their place in the ant colony. What makes a queen, what makes a drone, and they're able to alter the genetics of this, and you're actually able to alter the genetic structure and the behaviors of these plants. So to give you a sense of the kind of experiments that are being done at this time, so here you have a mouse, and the mouse is carrying a mutation in its genes that prevent its liver from metabolizing tyrosine. And tyrosine is just a molecule. We don't have to go into the details of that, but if that molecule builds up, it becomes toxic, and if you're a baby, it makes you sick. And so what we're able to do now is and scientists have done this, they've introduced CRISPR, the editing machine, into the livers of these mice, and it has been able to, what that CRISPR machine has done is that it's replaced the mutant defective form of the gene, and we've been able to, quote, cure this genetic disease in mice for the first time. And so you now have mice who have been CRISPR'd that are able to, who have recovered the function in their livers to metabolize tyrosine. Now it's really interesting because many people don't realize that CRISPR is also being used not only to genetically engineer individuals, but to genetically engineer populations of animals. So this is called a gene drive, and let me explain what is going on. So hopefully if you remember back from whatever bio you took, there are Mendelian rules that determine how you inherit genes from your parents. And one of those basic rules is that if you've got a gene, for example, this gene that makes this insect red, there's only a 50-50 chance that that gene will be handed on to one of its children. The reason being that, well, you have two copies. So if one of them is red, the other one is blue, there's a 50-50 chance you can give your kid the red versus the blue. But what we can do now actually is we've used CRISPR, and in this gene drive, what it does is that the version of the gene that you want, when introduced into the gene drive, actually converts the other copy as well. So both of your copies are now mutant. And what you can do is that you can put this insect into a population, and it starts mating with wild animals, and you end up having a situation within several generations where all of the insects in that population are carrying the mutant gene. Now, this technique is being used primarily to limit the number of mosquitoes out there. So you have Zika virus, you've got a whole bunch of different diseases that can be transmitted by mosquitoes, and what you do is, the goal here is to eradicate one of the 3,000 species of mosquitoes. We're not eradicating all mosquitoes. There's also only one mosquito species, Aedes aegypti, which is notorious as an insect vector. It's notorious for spreading a whole bunch of nasties. And the goal here is that people are trying to figure out if they can knock out this particular species of mosquito. Now, there are a lot of ethical questions that come up with this. You know, is it possible, should we be able to destroy, eradicate, and complete species of mosquito? Now, my students get confused because they go, we've never done this before. Well, actually we have, because you and I don't worry about smallpox, because we have actually eradicated an entire species of virus. We're in the process of trying to do that with polio, and hopefully with the coronavirus in Wuhan, we will eventually be able to eradicate that. And no one really complains about the absence of smallpox today. And I don't think people are going to be crying about the eradication of polio. But there's a question about whether or not there's a fundamental ethical difference between a virus and a mosquito. So there are experiments that are underway. So in November 2008, this is Anopheles gambiae. They've been able to use the CRISPR editing machine to begin the development of techniques to eradicate populations of mosquitoes. And they've actually been doing this. So they've not used CRISPR, that there are other methods where in the Oxitec Inc., this is a company that has developed a similar strategy for gene drives, and they have released their genetically altered mosquitoes in the Cayman Islands. And what you see there is you don't necessarily eradicate the entire species, but you severely reduce the numbers of individuals of infectious mosquitoes. So this is a very big question. We've also done this now with rodents. And so the question here is, a lot of people are excited about the possibility because they're very concerned with chemical pesticides. And they're saying maybe we'll just get rid, we will forego the chemical pesticides, which are very nasty, poisonous, toxic to the environment, and we'll go ahead and use germline gene therapy with rodents and gene drives to eradicate rats in New York, for example. The question is, how do we deal with this type of technology within an ecological account of the world, right? So you're going to try to remove all the rats in New York. How is that going to change the ecosystem? We don't know that yet, and so there are ethical questions that arise from that. Now, I'm going to spend the rest of my time talking about human gene editing, and there are two basic ways that we can use CRISPR at this point. You have what is called ex vivo down here and the in vivo. So let's talk about ex vivo. Ex means out of vivo life. So what happens here is stem cells are taken out of the patient. The stem cells are genetically edited outside the patient in the lab. Quality control is undertaken to make sure that those stem cells have been edited properly, and then they're returned into the patient in the hopes that this will cure the patient of some genetic disease. This is called ex vivo. There's also in vivo, where what you do is you take viruses and you use the virus as a delivery truck of sorts. You take your editing machine, you put it into the virus, you use the virus to infect the patient, and what happens there is that the virus introduces the editing machine into the target cells, and they get fixed within the patient. We're going to go through a couple of examples of that. So sickle cell disease, this has been done actually. So what you have here is that you have a single letter change. You have a G, A, so you and I, if you and I don't have sickle cell, you have a GAG in your genes. For those patients who have sickle cell disease, instead of the A, they have a T, and this is a gene in the molecule called hemoglobin, which carries oxygen in your blood. And for reasons there are reasons I can't get into at the moment, because of this single letter change in the DNA, the shape of the red blood cells changes and you have disease. And the idea here is that we will take out stem cells from your bone marrow, correct the GTG back into what it should have been, GAG, take that bone marrow, put it back into your body, and you hopefully will be cured. This is actually being done already, so less than a year ago, you had gene editing of human hematopoietic stem cells. Hematopoietic means blood-forming stem cells. The goal here is to genetically edit these and return them to the patient. Now, you also have in vivo gene editing at this time, and believe it or not, one of the target organs for this is the eye. The eye is an amazing organ to do these sort of experiments with. First of all, it's easily accessible. You can take a needle and inject stuff directly into the eye. Now, of course, it's a little gross, right? People get a little bit shocked, But when you have a age-related macular degeneration or you have an inherited blindness, the goal here is to try to save whatever sight you have by correcting the genes that you have, the defective genes that are destroying your retina. And so the idea here is we can either directly inject the CRISPR machine into your eye, or you can, again, package the CRISPR editing machine into a virus, which you would then inject into your eye. And so these, again, this is being done, so this is a couple of years ago. It's being called genome surgery, where you take the CRISPR-Cas9, and in this case, you're trying to treat age-related macular degeneration. And I work with a couple of the CRISPR companies in Cambridge, Massachusetts, and the eye is one of the target organs for this at this time, and there are clinical trials that are being undertaken right now in Cambridge and in Boston to test the following, to try to cure the following disease. So this is inherited blindness, it's LPA10. And all I'm going to tell you is there's a single genetic error in the genes of these patients. And what this company is trying to do is they're injecting CRISPR into the eyes of the patients so that you will correct the defect. And what will happen is in patients who don't have the correction, the cells that receive light, called photoreceptors, they're defective, they die, the patient becomes blind. The idea here is that we're going to be able to fix the genetic defect so that the remaining cells that are able to detect light don't die. And it turns out you only need about 10% of your retina to see. You're not going to be doing 20/20 vision. You're not going to be, be a sharpshooter. But one of the things when you meet these patients is you realize there's a profound difference between light and darkness and blackness. And for patients who are struggling with this sort of disease, the very hope of being able to see light and dark in order to get around your apartment without the aid of someone else is a profound improvement in quality of life. And so that's what they're trying to do, is because these are patients who have already lost a substantial amount of their sight, but they're still able to see something. We're trying very hard to prevent further loss of those retinal cells. Now, in 2017, this is Shukrat Vitalipov's work in Oregon, and this is the first series of experiments where human embryos were genetically altered using CRISPR. And this was the experiment that was done. So actually, what happened is they were able to obtain healthy eggs. Now, eggs are incredibly expensive, human eggs. So if you go online, you can go shopping for human eggs online. They range about $10,000 to $20,000. If the woman is tall, white, blonde, and went to the Ivy League, it goes up to $30,000, $40,000. And there are actually stories of undergrads who have paid their way through college, 1 egg per semester at a time. Now, sperm, cheap. Okay, sperm's like nothing. But with eggs, it's really expensive. So what they were able to do is they were able to get a healthy eggs, human eggs, and what they did is they fertilized the eggs with sperm taken from a man who had an inherited genetic disorder that affected his heart. And so they created embryos that were carrying this defective heart gene. And at the same time, they introduced CRISPR, the editing machine, and they discovered, for the most part, that a lot of the embryos into which they had introduced CRISPR were fixed. So, and now it's controversial how this fixing happened. I'm not going to go there right now. There's a debate in the professional literature. People are not quite sure how it got fixed, but it looks like the correction of the defect was more than what you would expect by chance. And then 2 years ago, you had the first 2 people born with genetic editing, Lulu and Nana, and this is He Jiankui who announced this at the Second International Human Genome Editing meeting in Hong Kong just around Thanksgiving in 2018, created a big ethical scientific debate over that. What he did was he actually recruited couples where the father was HIV positive. And he was able to convince the couples to allow him to genetically edit their embryos, claiming that he could make their embryos resistant to HIV/AIDS. Now, for background information, you may or may not know this, that 17% or so of people who ethnically are from Northern Europe carry a mutation called CCR5 delta 32 that makes them immune to HIV. They can get HIV, but they will never get AIDS. They're called elite controllers. And one of my students happened to do this. He happened to discover he was, so he ended up working at an HIV lab at MGH, because then he told his mother, I'm never going to get it, so I'm not worried. So the idea here that He Jiankui was basically trying to argue that he would recapitulate this genetic mutation that was in the population and introduce it into the offspring of these couples. Now, one of the things he probably didn't tell them is that the technology is not quite ready for that, right? And so he's currently in prison for 3 years in China for this. This is actually pretty light. For a long time, people thought he was going to be executed for this as a capital crime. So the 3 years is actually, in a sense, much, much lighter than a lot of scientists had worried about. All right. Now, this is October 21st, 2019. So just a few months ago, you have CRISPR 2.0. It's called prime editing. And this was a paper published in Nature. And the improvement of this technology, and I'm just going to summarize, is that it's able to edit the genome without cutting the DNA. Remember how I told you the original CRISPR machine would have to cut, and I also told you that cutting that DNA triggers cancer, which is not a good thing, right? We've now been able, this was an experiment, these were technology developed at MIT. What we're now able to do is we're able to edit without triggering that cancer process. And the hope here is that the error rate, you know, you want to change letter G on page 4 and 42, you don't know if you will accidentally change letter C on page 2. And so the error rate is significantly lower in this version of CRISPR than the original version. And so the hope here is that we're on the cusp of inventing a molecular editing machine where the risk of error is substantially low. It's low enough that patients would be willing to take the risk given the benefits that they would accrue. All right, let's talk about the ethics. And I'm going to talk about the ethics to compare and contrast the Catholic Judeo-Christian approach with more secular approaches because we live in a pluralistic society. So you get a sense of the debate that is ongoing. So it's really interesting that if you wanted to pinpoint one particular ethical principle that guides this entire debate, it's going to be the following. It's human dignity. And we talk about it a lot. It's in the Declaration of Human Rights from the United Nations. But here's the question. And this is the Catholic Church. So Dignitas Personae is a document, it's a bioethics document from the Vatican, and this is the first line. The dignity of a person must be recognized in every human being from conception to natural death. Now, a lot of people think this is a very theological statement, but if you look at the first line of the Constitution of the Federal Republic of Germany, it's called the Grundgesetz. The very first line is human dignity shall be inviolable. Now, historically, their constitution was written at the very end of World War II, and so not surprisingly, given the recent history with the Nazi Holocaust, they wanted to enshrine the protection of human dignity at the very heart, the very core of their constitution. Here's the question: what is it? That's the debate. And I'm gonna propose to you that a lot of the bioethical debates today is because we are not sure anymore what this is about. And when I teach this at the university, I say it's this question: how much are you worth? And so I say, Father, how much are you worth, Father? You know, and it's interesting. If I ask my students how much they're worth, they don't know how to say that. And so I go, all right, how about this? How much is an iPhone 7? I have to update the slide, but iPhone 7, because I teach undergraduates, they say you have to specify the memory. So 32G, how much is it worth? Now it's interesting because there are actually two values associated with it. You can determine the worth of that gadget in two ways. So if you are Huawei, or if you are Sony and you are a competitor of Apple, what you will do is you will buy one. You will go to Best Buy, you will go on Amazon, you will buy an iPhone 7, and then you will tear it down. You will take it apart and you will cost it. You will ask, how much does this phone cost knowing all that I know about phone parts and what is required to put these parts together. $219.80, plus or minus $5, because different people will estimate different ways. Now, of course, you have the suggested retail price, $650. Now, I want you to know that this is the intrinsic value. In other words, this is the price of that phone as soon as you put that phone together. You have not yet turned on the battery, you haven't turned on the phone. As soon as it's assembled, that's what it costs. And that's what it will cost until it falls apart. That's its intrinsic value. This value will depend. Is it Black Friday? Is there now an iPhone 28? What will happen is all of these external factors will alter the price based on market dynamics, pretty much. So you have an extrinsic value that will change depending upon time and circumstances and who is making that judgment. Now, faith-based traditions, including the Catholic tradition, claim that human persons too have both intrinsic and extrinsic worth. So if you talk about intrinsic worth, if I ask your mother, how much are you worth? Hopefully she will say, and my students' moms will say, priceless. Beyond price. And Immanuel Kant, the great German philosopher, would say, human beings do not have price, we have dignity. And this is at the heart of the claim that we are all equal. Now, if you think about it, and this is the argument of Peter Singer, every time you compare two people, you are always going to find some trait where one is more or less than the other. And if you do this over a population of 7 billion human beings, it is very hard to claim that we are all equally worth. Now, I grew up in Thailand, and one of the things that's really striking in a Buddhist society is that you do not have this inherent claim that human beings are intrinsically valuable, and that value exceeds monetization. And so you have a situation where it's the inherent worth of the human person is priceless. Now, extrinsic dignity, you have salary, you have net worth, celebrity, high office. I always tell my students, Kim Kardashian. Why? Why is she worth so much, right? And now, it's really interesting because in bioethics we can ask this question. If you have, let's say, the Prime Minister of Canada, and you have father, and they both go in and they both need a heart transplant, and you only have one heart, how do we decide which one gets it? Now, if you base it on extrinsic dignity, if you base it on salary, net worth, celebrity, or high office, father has a higher office, the world doesn't see that. But what actually happens is you will see is that the Prime Minister will, in fact, inevitably trump most of us in this room, if not all of us in this room. But bioethicists will argue that because we are inherently equal, the sickest gets the heart. Now you will see that, so when you go in there and you're on the waiting lists are based not on who you are, it's based on the need you have. And that is founded, the presumption there is we're radically equal. And the question now is, well, in a Christian society, in a Judeo-Christian Western society, it is based on this intrinsic dignity. The question is that our society, as a post-Christian secular society, they're struggling with this. Because you see, here it is. It is the worth of the human person simply because he or she is human. The phone is worth $218 because it's a phone, nothing else. You haven't turned it on, that's what it is. It is not earned or bestowed. It is acknowledged and recognized. That worth is just there. And you notice it's absolute. You cannot have half intrinsic dignity because there's no such thing as a half human being. Either you're human or you're not. And so that value, that intrinsic value, is immense. Now for Christians, and I would say for Jews, and as well as for Muslims, the intrinsic dignity is grounded upon the theological claim that human persons are made in the image and likeness of God. Because God is invaluable, he's priceless, because we are like him, we too are priceless. So this is a profound Christian account. But you see, it's really interesting. You don't have to be a Christian, Jewish, Muslim believer, as long as you're able to acknowledge that, and I have no time to jump into this, that because you and I have distinct ways of thinking, abstraction, conceptualization, we have an immaterial component to us. Immaterial is always worth more than material. My students go, why is that? I go, put it this way. You have two iPhones. One has an expiration date and one doesn't. Ever. 5 billion trillion years. Which one is more expensive? And they go, of course, no expiration date. So the immaterial, if we are in fact immaterial, then, and we all are immaterial in equal ways, then we are worth more than any iPhone or any other creature that you see. Now, in a culture today that denies, first of all, the Christian gospel, and then is radically materialistic and reductionistic in the way it looks at the world, neither one of these options is available as support for the intrinsic dignity. So we now are living in a society where everything is extrinsic dignity. And so now it's about who gets to decide how much you are worth. So if you decide you are not worth enough, you should be able to euthanize yourself. You see, it's about deciding your worth. So faith-based bioethics is grounded on this idea that it's both intrinsic and extrinsic human dignity that have to be supported. Now let me just quickly apply this view to CRISPR. So how do we understand this? So there are dignity concerns for human genome editing. You can divide them into 4 different categories. The first is safety of persons. If people have this inviolable, unmeasurable, inestimable, beyond price value, then you have to make sure that anything you do to them respects that and doesn't undermine their life. And so you have safety concerns, off-target mutations, and I'm suggesting to you that technology is accelerating enough that we should expect that within 10 years or so, we should be able to drop that error rate below what most people would say is a reasonable threshold for risk. Chimeric tissues, this is the question of, well, if I'm going to inject it into your body and I'm only going to edit 20% of your liver, is that going to be a problem? Because 20% of your liver is edited, but 80% is not? I told you in your retina, as long as you have 10% of edited retinal cells, you'll be able to see. So there's a lot of questions of mixed tissues, edited and non-edited. That's called chimeric. This is the commodification of persons. This is the question of designer babies. If you decide to design a baby, the baby becomes a means towards your dreams. It's a means towards the idea here is that you've commodified, you have made that baby a means towards your fulfillment rather than looking at that baby as an ends in himself or herself. And the question here is that when you are commodifying people, you are attacking that dignity because you are treating them like property. And one of the things about human beings is you can never be treated as property. That's really striking because I teach moral theology as well. And last week we had a discussion about slavery. And I asked my students this, should a man be allowed to sell himself into slavery in order to save the life of his dying mother who needs a lot of money for this incredibly expensive drug? More than 2/3 of them said yes. We are in a world where even slavery is confused. They think that as long as you decide it, you can be a slave. And I told them, first of all, we live in a country, there's the 13th and 14th Amendments, it's illegal. And one of my students said, well, that will change eventually. It's an interesting view. He said, we now know that slavery, as long as you give consent, should be fine. Because consent is what drives ethics. And you see, It's consent of worth. If you decide you are not worth what we think you are worth, you can do whatever you want. It's all about consent. There's also marginalizations of persons. There's eugenic temptations, right? One of my students actually said, Father, you know, what's going to happen in 100 years is all the Jews and the Christians, they're going to be the ugly ones. Because all the secular people are going to genetically engineer their kids, right? Now here's the question. This is the problem of eugenics, though. Eugenics is the temptation to think that imperfect means inhuman. And that's the danger in a world that's seeking perfection, is we reduce those so-called imperfects to the inhuman category. The other issue, too, is that the poor get poorer and the rich get, I mean, this is incredibly expensive. So what actually happens now, you know, you go to Manhattan and you have families where they're paid, as soon as the kid is born, the family is actually paying for kindergarten, so that they go to this amazing kindergarten, so they go to that college, that. Can you imagine if those families had access to genetic engineering where they could give their kids, they can make their kids tall? Everyone knows, there's a lot of studies show that taller people have more opportunity than shorter people. But as I point out to students, just being tall doesn't mean it's always cool. One of my Dominican brothers is 6'5", he's great, he's tall, but when he flies to Rome, it's incredibly painful, because especially now, I mean, regular economy is impossible. Economy plus is not that much more, because they are just cramming us in there, the sardines. So there's always advantages and disadvantages of everything. And finally, of course, especially for Christians who the Lord has called us to always look out for the poor and vulnerable. So what I'm going to do now at the very end is to talk about how, a framework for using CRISPR with humans, and I'm going to highlight two different distinctions that you need to know. First is therapeutic versus non-therapeutic. So therapeutic here is preserve and promote health and well-being. Non-therapeutic, I define this as to preserve and promote personal and social opportunity. And you know what I mean. If you want to design your kids, your kid is 6'4", you're going to say, well, I want him to be president of the United States. That's opportunity, that's not health and well-being. The other distinction is this: somatic gene editing and germline genetics. So somatic gene editing is, I modify the genome of a living human organism, you or me. That's called somatic gene editing. Germline gene editing is to modify the genome of a future human organism, a kid, a child. And so I'm going to basically lay out a proposed ethical framework in this way. You have therapeutic and non-therapeutic on this end, and then you have somatic and germline on that. And I'm going to propose the therapeutic, somatic is fine, everything else is not. Let me go try to explain some of this. So this one, as long as it's safe and effective, we should use gene editing to alleviate suffering, cure disease, and to protect health and well-being. It's just like any other medical intervention. Now, a lot of people will say, if I can fix you, why can't I fix you so that your kids don't get the disease? So this is the germline gene editing therapeutic, and I'll say my opposition to this is actually different. I'm arguing that we cannot do this simply because we can never know the risks and benefits of this unless we do it. So for example, I have a new drug, and this drug is supposed to be the wonder drug against prostate cancer. What I do is I say, all right, we need a phase 1 clinical trial. Are there patients out there who are dying of prostate cancer, and we tell them, look, this drug may help you. So they volunteer, they give informed consent, we give them this drug, and we can see whether or not there are risks, what the risks are, and what the benefits are for that drug. And if the risks are minimal, we move it to phase 2, then phase 3, looking for efficacy. Now, you can't do that with germline gene editing of babies, because there are no germline gene edited babies. So in order for you to determine if it's risky or beneficial, you would actually have to do it. And there are ethical principles that prevent us from simply doing ad hoc experiments in humans The Nuremberg trials highlighted that. You cannot just do experiments, that we have no idea about the risk and benefit. So I'm saying no to that. And then there's this no here, the non-therapeutic, and this is the last slide basically, for the following reasons. This is taller, shorter, smarter, more beautiful. Okay, there's social stigmatization and discrimination. So as people become more beautiful, the ugly, the rest of us, right, we're left behind. Then there's social pressures for parents. So parents now feel the pressure. And what's really interesting here is that I end up focusing on the kids. There's what is called the open futures argument. This is the idea. If you design a kid to be Kobe Bryant, may he rest in peace, right? And the kid shows up and he is Kobe Bryant Jr. And he says, look, I want to be a piano player. I've got such a large, amazing hands. His parents are like, we designed you to play basketball. So this is called, you close the futures for the kid. And my students understand this, because I tell them, how many of you are under pressure to go to medical school? And they're like, you know, they want. Imagine if that pressure was actually encoded in your genes. You knew from the moment that you were born that you were meant to be designed for this versus that. The kind of pressure that would put you in, especially if you didn't have any say. Right? And so here, the idea is that genetically edited children will face detrimental psychological and social pressures as well. All right, so just to close, to acknowledgements, this is my laboratory. I have an amazing laboratory of undergraduates. So I have an undergraduate university, and Providence College is a Dominican university, so my religious order ministers and runs the university. And so they have, even my science majors have a very robust liberal arts, humanities background. So they all have read Brothers Karamazov. They've read The Plague by Camus. They've read Aristotle, Nicomachean Ethics. And so what happens is late at night, often late, I don't know why, but always late at night, we're running gels, we're doing experiments, and they will start talking about life. And they start talking about, and so I always thank them because they're the ones who ask me about CRISPR, what about a kid's father, grandkids, so they challenge me to think about these questions in a way that makes it clear how God has called us to be the kind of people we are called to be in the 21st century. And that's my funding for my research laboratory. Thank you so much.